The non-specific part of your immune system is the part that works no matter what you've been infected by or what's caused the damage. So, for example, clotting, antiseptic tears, inflammation, fever, phagocytosis. Those things are going to happen no matter what's causing the problem. The specific immune response is targeted at specific antigens, and the B lymphocytes and T lymphocytes work together in order to cause these responses to occur. Let's look at the key differences between these two types of lymphocytes. First thing, they both are made in the bone marrow, but the B lymphocytes mature in the bone marrow and the T lymphocytes mature in the thymus gland. The B lymphocytes respond to antigens which are outside of our own cells, i.e. bacteria and viruses which are just floating around the body. The word humour is a very, very old medical term that describes fluid, like tissue fluid or blood. So your B lymphocytes are involved in humoral immunity because they're responding to things that aren't locked inside your cells, they're just floating around your body. T lymphocytes respond to antigens which are on or inside our own cells. So, for example, sometimes an infected cell, because of the damage that is happening within that cell, the antigens on its cell surface membrane will change, and T lymphocytes will respond to that. So T lymphocytes will respond to cells which have been altered by infection. If you have a cancer cell, then that cancerous cell is likely to have lots of mutations to the DNA, but the cell is still undergoing mitosis even though it's not meant to be. And because of all those changes, the cell's antigens are going to be different to a healthy body cell, and your T lymphocytes will respond to that. And if you have to have a transplant, whether it's a blood transfusion or, say, a kidney transplant, if the antigens on the outside of those cells don't match or they look foreign to your T lymphocytes, they'll respond to those as well. So your T lymphocytes are involved with cell-mediated immunity. They will respond to antigens which are on a cell's surface membrane, not a bacterial cell, but a human cell. B lymphocytes are the ones that make antibodies. T lymphocytes help to get rid of that infection by making other kinds of cells. This flowchart shows cell-mediated immunity. Now, in this case, this very first cell is a phagocyte. Specifically, it's going to be a macrophage because it's ingested a pathogen, it's digested it down, so you can see it's broken it up into its constituent components, and then it's attached the antigens onto some glycoproteins which are floating around the cytoplasm to make MHC, major histocompatibility complex. That molecule is then presented on the outside of the cell's surface membrane, i.e. this macrophage has become an antigen presenting cell. It's one of our cells that now has different antigens on it, so it's cell-mediated immunity. This process would also work if we're talking about infected cells or cancerous cells or transplanted cells. This is one out of maybe millions of different types of T cells, and each type has a different shaped antigen receptor on the outside of it. So initially we are waiting for the correct T cell which is specific and complementary to this antigen, where the shapes correspond with each other, they match together. We're waiting for the right T cell to come along and bind onto this antigen, which will only happen if the shapes are complementary, and that will activate this T cell, and that's called clonal selection. One activated T cell by itself cannot actually do very much, so the next thing you need to happen is clonal expansion, which is where that single cell is going to undergo mitosis to make lots of copies of itself. Now, when you did the cell division topic, you were taught that typically, once a cell has been differentiated, it's not going to undergo mitosis again. Undifferentiated cells, like stem cells, keep going through mitosis. There are some exceptions to that. Liver cells are an exception to that. You can lose quite a lot of function of your liver, but the remaining cells are able to undergo mitosis to replace and repair the damage. and B cells and T cells are also exceptions to this. These T cells, once they've carried out mitosis, are going to further differentiate. They can differentiate into T killer cells, and these T killer cells are then going to go back onto the original infected cells to kill them. And the way that they do that, well, one of the ways they do that is they produce a molecule called perforin, which perforates the cell surface membrane. It pokes holes in maybe the infected cell. When it pokes holes in it, the cell surface membrane is now freely permeable. So lots of water is going to rush in. And because lots of water is rushing in and these cells don't have a cell wall, the cell will explode. It undergoes lysis, it destroys the cell and the pathogens which were inside it. The other kind of cells that are made are T regulator cells, also called T suppressor cells. These work to inhibit the immune response so that your body doesn't keep wasting resources making more and more T cells or B cells when it's not actually needed. So it inhibits the immune response when you've already dealt with the problem. You also get T memory cells, so that next time the same pathogen comes into circulation, instead of waiting for maybe 1 in a million complementary T cells to become activated, now maybe you've also got 1,000 additional complementary memory cells. So activation will happen faster, expansion will happen faster, and you'll get quicker T killer cells being made to get rid of the initial cells. That means that the pathogen is destroyed before it hopefully has a chance to cause too many symptoms. And finally, you have the T helper cells. And what the T helper cells do is they are needed here. So that's the T helper cell that was formed in the previous flowchart. Now, they're needed as part of the humoral immune response, which means that these two processes are not completely standalone. They do work together. Now, if it's humoral immunity, we're talking about B lymphocytes. This is a B lymphocyte. And similar to T cells, there are millions of different types of B cells, each of them with different complementary receptors on the outside. Now, this is not phagocytosis, although I appreciate it looks like that. What happens is if this B cell comes into contact with antigens like within a toxin or like the ones on the outside of a bacteria, it comes into antigens that it's complementary with, then it carries out endocytosis, brings those antigens within it and presents them on its own cell surface membrane. So B cells, once they've taken in antigens that they are complementary to, also become antigen presenting cells. This is the same B cell. It's not a different cell. And diagrams showing humoral immunity often show them in this way like flowcharts, and that tends to confuse people. So this cell number 3 is the same B cell that is now presenting its own antigens. In order to become fully selected and activated, it will only undergo mitosis if the T helper cell will also attach onto it. So the T helper cell helps to activate the B cells. And again, one of the theories about why this happens is to make sure that your immune system doesn't overreact. It doesn't need to use resources to make more cells and make antibodies if the pathogens have already been dealt with. Once these B cells are activated, they differentiate into 2 main types. The first one, plasma cells, which produce the antibodies. You did a unit of work on antibodies looking at their structure. Think about how they work. They work by causing different pathogens to agglutinate, to clump together so they can't be as active. They work by marking them, because when the antibodies are attached to the pathogens, it makes it easier for phagocytes to come along and engulf and digest them. Sometimes they attach onto the toxins, which means that they neutralize the toxins as well. So the antibodies and the T killer cells provide the short-term solution to get rid of the pathogen or neutralize the toxin. The memory cells, so here you've got B memory cells, provide long-term immunity. so that the next time that same pathogen comes into your body, you already have a lot of complementary memory cells which are also potentially activated. They will be activated faster than the first response, which means that more quickly you get antibodies being produced before that pathogen has a chance to actually cause the symptoms to make you ill. So it doesn't mean that you won't get the same pathogen inside your body. It just means that when it comes again, it won't have a chance to actually cause the symptoms.